three people who are going to be leading the presentation to get us started. We have Christina Warrener, who's an associate professor of anthropology at Harvard and is also a group leader of microbiome sciences at the Max Planck Institute for Evolutionary Anthropology. She also holds a professorship at the Friedrich Schiller University in Jena. She and her research team are doing amazing work studying the relationship between humans, their food, and their microbes. And importantly, this also includes traces of food and microbes in the archaeological record. With her is Irina Velsko. She's a postdoctoral researcher at the Max Planck Institute for Evolutionary Anthropology, got her undergraduate degree at UCSB and a PhD in Florida, at University of Florida. And then we have Alex Hübner, who's a postdoctoral researcher also at the Max Planck Institute in Leipzig. He got his undergraduate degree, a master's degree at Berlin, and a PhD through the Max Planck Institute International Research School. Okay, so I understand that Christina is going to do most of the presentation and then open it up for discussion. We'll be able to talk with all three of the authors on these papers. So go ahead, Christina. Welcome to Club EvMed.
Thanks so much. I'm just going to go ahead and share my screen. Let's see here. Can everybody see that okay? Great. Well, thank you so much for inviting us to come and share some of our research here. It's a real pleasure and an honor to be able to have this conversation. What I thought we would talk about today is some of our ongoing research that we're really excited about. A lot of the work we do in our group focuses on the long relationship that humans have with their food and also with microbes. And in particular, we become really interested in specific modes of evolutionary adaptation that humans have to different types of sugars. And today we're going to focus on kind of two. We're going to focus on amylose and amylopectin, so starches, and also lactose in milk. And this is going to feature a lot of the work that Irina and Alex have been leading in our group. So one of the things that I find most interesting about humans is just how incredibly varied our diets are. One of the things that characterizes us is that we are dietary generalists. We can survive on a wide variety of foods. And one thing that I think is also really interesting is that much about what we understand about how we became human also relates to food. So there's certain changes within our food choice and food selection that seem to be really critical for the emergence of humans and for Homo as a whole, as our genus. And so what we're going to do today is kind of revisit some of these classic examples of that are noted as being major milestones within human evolution as they relate to food, and how by studying the microbiome we might be able to get extra insights into this. So today I'm going to focus on kind of two really classic examples of how the diet has played a major role in human evolution. These are textbook examples, one being the expensive tissue hypothesis, which is an idea to try to explain how Homo emerged, how we grew our large brains, and how we solved energetic problems to do so through changes in diet. And the second one is focusing on the gene culture hypothesis of dairying and taking a closer look at lactase persistence. Lactase persistence is the genetic trait that seems to be under the highest selection in the entire human genome. Do we really understand this and how people are adapting to dairying? So we're going to relook at some of these older questions using new methods and see if we can understand them a little bit better by taking into account, for example, the microbiome. So just as a recap for those of you who aren't familiar with the expensive tissue hypothesis, this was first proposed in 1995 by Leslie Aiello. This is a really interesting idea. It came out of an observation that she made that over time, what we see is a massive expansion of the brain within Homo. And really after about 1.9 million years ago, we start to see this rapid growth of the human brain. What is puzzling about this is that at that same time, over that same period, we see a reduction in the size of the gastrointestinal tract. So we have this kind of paradox where we see greater need to grow this very energetically costly and expensive brain. At the same time, we're shrinking the size of our gut. And so she says that the only way to solve this paradox is that there must have been a dietary shift or change that occurred such that we switched to a higher quality diet, which allowed us to shrink the anatomical and physiological need for digestion and maintain our large brain size. So within this, though, so I think there's a widespread agreement that there was a dietary shift, that it must have accompanied this growth of the brain. But what is that dietary shift? And there's been a couple of major hypotheses for this, one being the kind of raw meat hypothesis, which was proposed as an explanation by many people. Catherine Milton wrote quite a lot about this, and really arguing that an increase in availability of raw meat through cooperative hunting may have driven this, and this may have also explained the need for a larger brain and more cognitive ability. An alternative hypothesis is one that was proposed by Richard Wrangham, which I'll call here the Cookivore hypothesis, the idea that actually what may have driven some of this increased nutritional density may have been cooking, and in particular, cooking starches and underground storage organs like roots and tubers may have provided the glucose that was needed to grow this large brain. So what do we know about these hypotheses? These two hypotheses have been proposed for a long time, and there's evidence in favor of both. So for the raw meat hypothesis, I mean, first of all, we know that early Homo and consumed meat. We see lots of evidence of this in the archaeological record, paleontological record. We have lots of evidence for cut marks on bones. We have faunal remains scatters at Neanderthal sites, for example. We know that animal products played an important role in the diet. But one of the problems we have is that our knowledge of the plant components of diets is strongly biased because plants do not preserve as well in the archaeological record. They're largely invisible to us. And so what we need is a method to be able to kind of weight the relative importance of animal products in early Homo diets. And one of the ways this has been done is through looking at stable isotopes of bone collagen. This is something that Mike Richards was really a pioneer of, looking at, for example, Neanderthal diets to try to understand the relative importance of animal products versus plant components in the diet. And specifically by looking at the heavier isotopes of nitrogen, you can make some quantitative inferences about the relative proportions of the diet from different classes of food at different points in the trophic levels. So here you have basically the heavier isotope of nitrogen enriches as you go up the trophic level, and there's about a 3 per mil increase with each step up the trophic level. So the delta N15 of plants is relatively low, it's higher in herbivores and then higher in carnivores. And the idea is then you could measure bone collagen of humans or Neanderthals to try to estimate where they fall within a food web. This was applied to a number of samples throughout Pleistocene Europe, and very interestingly, humans and Neanderthals fell at the very top of this food chain in this sort of reconstruction, in some cases even exceeding that of wolves and hyenas, and this was largely argued to be evidence that, that Neanderthals and early humans were top-level carnivores. But there are problems with this. And one of the major issues is that the isotopic ecology of the Pleistocene is extremely complex. And we know this from many lines of evidence, and there's a lot we don't really understand. We know that you can't just take Holocene models and project them backwards, because we see all sorts of strange effects like this one, where mammoths that have been measured over time actually span all 3 trophic levels, and in fact are sometimes almost as high as the highest carnivores from the same time period. And we know there's never been a carnivorous mammoth. So something is very strange that's going on during the Pleistocene in terms of isotopic ecology. This may have to do with periodic fasting or consumption of detrivores, which can also alter the isotopic ratios. So there's still a lot of questions as to the actual proportion of animal products that were important in early human diets. On the Cookivore hypothesis side, there's a lot of really interesting evidence here. Until fairly recently, it wasn't clear how far fire went back, and it was thought it was relatively recent. And if so, that would put it too recent to be a major player for the increased size of the human brain. But some more recent research by Sarah Hlubick and others is increasingly pushing back that fire record more and more. So there's now some really compelling evidence that controlled fire goes back to at least 1.5 million years ago, which is getting us closer to that 1.9 period where we see the really large growth of the brain. So this is an ongoing area of research. It is really hard to find evidence of fire in the archaeological record. There's a lot of processes that work against the preservation of fire remains. But by being really creative about using multiple lines of evidence, increasingly we are able to push this back further and further. And in particular with Sarah's work, really focusing on open air sites, which actually might represent the majority of sites in the past, we may be able to get a better handle on early cooking. There's some other great evidence for cooking as well. So this is work pioneered by Amanda Henry. looking at preserved microfossils of plants within dental calculus or calcified dental plaque on teeth. She found early on that there are actually preserved starch granules within Neanderthal dental calculus of a wide range of starchy plants, and this is really interesting. In particular, some of the starch granules that she found, when starch is packed into a granule, it's in a semi-crystalline state, but as it's cooked, it starts to gelatinize. And what she observed was that some of the starches that had been mineralized within Neanderthal dental calculus actually show evidence of this beginning stages of gelatinization, suggesting that they may be cooked starches. So this is some evidence on the kind of the side of the cookivore hypothesis. We also know that early humans, as they were expanding into new niches, were starting to occupy different types of ecological zones where starch-rich foods are really quite abundant. And we know that almost nearly all hunter-gatherers today will collect starch-rich foods. We also can look at primate analogs who live in some of these similar environments, and they also will target starch-rich foods. So this does seem to be something that is becoming, that was available during early human evolution. And then there's also some really cool work that's been done by P.J. Perry and others looking at salivary amylase. So this is an enzyme that's expressed that can break down starch. We express it in saliva, and this is something that develops within the primate lineage. And one of the things that he found was that while chimpanzees just have a single haploid copy, we see expansion of this, so copy number variation within anatomically modern humans, and in some cases, really high numbers of copy. So this seems to be something that has occurred within humans, suggesting that there's a selection for increased capacity to digest starch. Omer Gokeman has also explored this beyond humans and beyond primates, looking at amylase gene copy number expansion, and has found that this is actually a pattern that has independently occurred many, many times within different mammals, and in particular mammals who have a broad range diet containing starches often show this copy number expansion in the amylase genes, whether in salivary amylase or pancreatic. And we see this in dogs, we see this in pigs, we see this in a variety of rodents, and we see it in humans. And so the argument that he's making here is that this copy number expansion of amylase is related to an adaptation of starches. So could this be an indication that humans were early reliant on starch? Well, one thing we can do is we can push this back into the archaeological record now that we have complete genomes for some Neanderthals and early humans. Could we actually ask how many copies of salivary amylase did they have? So there's only 2 Neanderthal genomes that have been sequenced deeply enough to determine copy number. And what we can see is that both of them have 2 diploid copies, or 1 haploid copy each. So they have that ancestral state of copy number for salivary amylase. But for the earliest humans that have been studied, again, there's not that many genomes that we have available, but for those that we do have, they all show a copy number expansion. So this is really interesting. So in this case, the Neanderthal seems to show the ancestral state with just the single haploid copy, whereas anatomically modern humans have this expansion. And so Thomas Kivisild and others have tried to model this, and they've really argued that this human amylase copy number sweep is specific to our species and is a very recent phenomenon. So kind of pushing it too far forward into the future to be relevant for the expansion of the brain. But how do we explain some of the results that were found by Amanda Henry and others of what appears to be starch reliance on earlier Homo? So this is where my group became really interested in this question, and working with my student James Fellows Yates, who's now a postdoc, and Irina Velska, who's with us today, we tackled this problem by trying to see if we could look at additional lines of evidence that might inform this. And so what we focused on was looking at dental calculus in a wide range of primates and also Neanderthals and early humans, and specifically looking at the oral microbiome. And could we see, were there any changes in the oral microbiome that might inform on this question about early human starch reliance? So what is dental calculus? It's calcified dental plaque. It forms incrementally over an individual's lifetime. You form a layer of plaque, it calcifies, a new layer forms, and it grows like rings of a tree or layers of an onion. The preservation within dental calculus is really extraordinary. We can zoom in here and see individual microbial cells that have been calcified during life. It's the only part of your body that fossilizes while you're still alive, and this leads to really tremendous preservation. We can decalcify that, and we can bind fluorescent dye to double-stranded DNA, and you can see we have enormous amounts of DNA preservation. In fact, it's the single greatest bioarchive of ancient DNA in the archaeological record is in dental calculus. And so we applied these techniques to try to understand the microbiome, the oral microbiome, and how it had changed. And what we found was that there's a relatively coherent oral microbiome across human evolution, some features that are quite distinct for Homo. And what was really interesting is that among the features that distinguish the oral microbiome of Homo versus other primates was in carbohydrate metabolism. So we see that there's a carbohydrate metabolism is one of the functions in which our oral microbiome differs from that of other primates. And we can take a closer look at that and try to investigate what are the pathways and what are some of the taxa that might be driving that. And one of the things we found is that most of that variation is actually explained by this kind of yellow group here coming from Streptococcus. Streptococcus is a really abundant and prevalent bacteria in the oral cavity of primates in general, and in particular humans. But at the level of genus, that's not very informative, because Streptococci have many different clades that have different functional niches and different metabolic capabilities. And so what we decided to do was look a little bit more closely at some of these functionally distinct groups to see if there were any patterns. And what we found was that we have a variety of different streptococcal groups within howler monkeys, for example, a little bit noisier within gorillas. Chimpanzees have a really unusual Streptococcus profile. First of all, the level of Streptococcus is very low, and they are completely dominated by the anginosis and the pyogenic groups. So they really have a very different consortia of Streptococcus. And then humans are yet different again, where most humans have a really dominant sanguineous streptococcal profile. This is quite interesting because the sanguineous, mitis, and salivarius groups, and in particular the sanguineous, have an unusual feature that is not shared by other streps or other bacteria. Today, these bacteria are known for having something called amylase binding proteins. This is a super cool thing where these bacteria have evolved to be able to capture the salivary amylase from our own saliva and to be able to use it for their own purposes to feed themselves. So they are unique amongst oral bacteria in being able to digest starch because they've stolen the amylase from our own saliva to feed themselves. There's two genes that are primarily responsible for this, the amylase binding protein A and B. And so we wanted to investigate these a bit further. What's also interesting is, in particular for salivary, for the amylase binding protein A, it's only expressed in the presence of starch. So it conveys no advantage if starch is not present in the diet. The two of these work together synergistically, and they give two advantages to these bacteria. They provide metabolic advantage because it allows them to gain nutritional benefit from starch, whereas no other oral bacteria can do this. And it gives them colonization advantage because salivary amylase actually is a major component of the acquired enamel pellicle, or this thin protein layer that covers the teeth and allows a bacteria to bind on and to colonize. So this gives a huge advantage to bacteria that combine salivary amylase for colonizing teeth and dental plaque formation. So this is really interesting. We know that that present day sanguineous group bacteria in humans have these traits, but we didn't know how far back these genes go back in time. And in particular, amylase binding protein A is a horizontal gene transfer. So we don't know when and where that first entered into human oral streps. So the first thing we did is we then looked for the presence of these genes within our non-human primates, and what we found is very little evidence for them. Most of the hits we have here are probably spurious, really little to no evidence for amylase binding proteins at all within the non-human primates. When we look at members of Homo, however, we see abundant evidence for it. And what was very surprising was we see it not only in anatomically modern humans, but we see it in Neanderthals as well. So this suggests that starch had already become important enough in the common ancestor of humans and Neanderthals to have selected for an increase in the sanguineous bacteria and for this particular gene. So this suggests that starches may have been quite important earlier on, and perhaps it's just the delayed effect of the expansion of the amylase gene copy number, or maybe there's additional pressures later on, but that starch may have indeed played a really important role. We're now going to be digging much more into this. This is something that Irene is really actively working on and trying to get a better sense of these genes. They're present in multiple species. They have different evolutionary histories, but we think this may be a key to understanding some aspects of early human diets. And so this is something that we're following up on. All right, now I'm going to transition and start to talk about lactose and the gene culture hypothesis of dairying. So milk is really interesting. I'm fascinated by the history of dairying because dairy is this, it's a truly manufactured food, and it's a prehistoric manufactured food, and it's a really unusual one. So global milk production today is about 697 million tons of milk, which is really hard to envision how much milk that is. Maybe some more familiar units is about 278,800 Olympic-sized swimming pools worth of milk are produced every year for consumption. It's enough for every human on Earth to consume a quarter liter of milk every day. And yet we think that the vast majority of the world's population is actually lactose intolerant. So this leads to a kind of question as to why we have developed this sort of globally important food that is not digestible by many, many people. And how do we come to have this really unusual food class that we call dairy products? So on the one hand, milk is the most natural food for humans. It is the food of infants for all mammals. And so it's really natural in that sense. It has also an amazing nutritional profile. It's rich in sugars and fats and proteins and minerals and vitamins. It's really a complete package of everything that a growing infant needs to consume. But it's really unusual. Humans are really unusual in the sense that we don't just consume our own milk, but we consume the milks of other animals. We're unique in that respect. And we're also unique in the respect that we consume milk long after infancy and even into senescence. So this really distinguishes us from all other mammals. And so why is there this barrier to consuming milk as adults in other species, for example? And the main barrier is lactose. Lactose is a disaccharide of glucose and galactose, and it requires an enzyme called lactase, which is encoded by the lactase gene on chromosome 2 to break it down. So basically it has a beta-glycosidic bond that is quite difficult to break, and it requires this enzyme to hydrolyze it so that the individual sugars can be absorbed. Lactase itself, the enzyme, is expressed in the small intestine, and this cleaves this bond so that the two monomers of sugar can be absorbed by the small intestine. And this lactase expression is controlled by this regulatory region located a bit upstream in another gene, in the intron of another gene called MCM6. And so what happens basically is that if you have lactase expression, what happens if you consume milk is you break this bond, you produce the glucose and galactose monomers, and those are readily taken up by the bloodstream, and it leads to growth. Alternatively, if you do not produce lactase in the small intestine, the lactose will travel undigested into the large intestine, where there are a huge consortia of gut microbes who are more than happy to digest it for you. However, as they ferment that lactose, they typically produce a really large amounts of hydrogen gas and other byproducts that contribute to the symptoms of lactose intolerance. So I mentioned before that, that the regulation of lactase expression is controlled in this other gene called MCM6. And basically what happens is during infancy, mammals produce large amounts of lactase. And then as an individual ages, that slowly reduces until almost none is produced. And this is just part of a normal weaning process of mammals in general. So how is it, though, then we have adult consumption of animal milks within human populations? How does this actually work? Well, there was an enormous amount of research in the early 2000s to try to track down the causes of lactase persistence, or this persistent expression of lactase into adulthood. And it was found that subsets of human populations had mutations in this regulatory region of MCM6, which effectively allowed the lactase to stay at full production for the entire lifespan. And these were found in different populations. So one of the genetic variants was common in Europeans, people in Anatolia, Iranian Plateau, northern South Asia. Other variants were found to be common in the Arabian Peninsula and also parts of North Africa. Other variants were identified on the Tibetan Plateau, and multiple variants were identified in East Africa and associated with pastoralists. And so by disrupting this regulation, then this led to lifelong high production of lactase, kind of simulating an infant-like state. And so at the same time that this research was taking place, this was also kind of really the dawn of ancient DNA research, and a lot of new evidence was coming out about the importance of the expansion of Neolithic farmers from the Near East into Europe. And this was really exciting. And a lot of researchers are starting to converge on this idea that these Neolithic farmers were spreading out. There was also evidence these Neolithic farmers were producing dairy products. This was measured by Richard Evershed and colleagues looking at dairy fats within pottery. And this was found both in the Near East and Europe. And so this kind of idea came together that this would have emerged, this idea that lactase persistence trait, at least the European variant, had emerged during this early Neolithic period, and that had reached its present-day global distribution through the migration of these farmers. And this idea was really synthesized in this article that was written in 2013 by Andrew Curry, where it outlined this whole model of the standard model we now have today of the origins of lactase persistence. It's spread with farmers throughout Europe to explain present-day distributions of lactase persistence and lactose tolerance. And so this was really exciting. And I remember when it came out, it just felt like, ta-da, we've solved this problem, this gene culture evolution question of dairy. But very quickly, there were major problems with this theory. The first one actually came around this time in 2007, when Joachim Berger and his colleagues performed the first study to try to really look for ancient DNA evidence of this lactase persistence allele in these Neolithic farming populations, and they didn't find it. This was a really inconvenient finding because it contradicted the model that had been built up until this point. But it wasn't a very large sample size. And so the idea here, well, possibly they just sort of missed it, or maybe it was just too low frequency still, it was still rising. And so it was largely kind of viewed as inconclusive. Fast forward about 8 years, and 3 very large papers came out with many hundreds of genomes. And it was very clear that lactase persistence was not present during the Neolithic. And in fact, the earliest evidence comes in during the late Bronze Age, and it really only starts to rise to frequency in the Iron Age. And so that's, it means there's a 4,000-year gap between when we have the earliest physical evidence for milking and dairying and the earliest evidence that now exists for lactase persistence in the archaeological record. So it's really clear now that lactase persistence did not arise amongst these early dairy farmers, and that Europe's cheese-making linear pancreamic or earliest farmers were not lactase persistence, and that we have this gap, as I said, dairying precedes lactase persistence by 4,000 years. How did this work? How do you get a dairy-based society if you don't actually have the capability to digest lactose? So this is where the kind of field stands now. What we do know is that lactase persistence is essentially irrelevant for the early history of dairying. And then just a few weeks ago, a new paper came out by Mark Thomas and Richard Evershed and other colleagues who had done a lot of this initial modeling, finding that it's only rising really quite late. And we're not exactly sure why. It could have to do with disease stress. There's a lot of hypotheses, but it's clearly not part of the initial dairying story. So this is where my team kind of comes into this. I've always been really fascinated by this question. And what's interesting is, although we have populations around the world who are dairying and who have lactase persistence, there's always been a few exceptions that never fit that model. And in particular is Mongolia. So in Mongolia, Mongolia has a dairy-based society. About 40% of the population today subsists on dairy products through mobile pastoralism. And today in Mongolia, they milk more species of dairy livestock than any other country on earth. They milk horses, camels, reindeer, yaks, cattle, sheep, and goats. And they produce a really wide variety of dairy products that are consumed, including fresh milks and yogurts and a whole variety of cheeses and butters and many different dairy products that we don't even have names for in English. And so we've been doing a lot of work, ethnographic work with mobile pastoralists in Mongolia to understand better the wide variety of dairy products that they produce, their lactose content. What you see here on the left is some tarag or yogurt and some ezgi, which is a type of like skim milk cheese. And then in the center here, you see Geril, who's showing us how to distill fermented yak yogurt to make a kind of yak milk a vodka. So it's a still. So a really wide variety of dairy products. So we've been trying to understand how we can have this situation in Mongolia where we lack lactase persistence today, and yet seem to have this really strong reliance on dairy products. We've done some work looking into the archaeological record. We did a really large study looking at 6,000 years of Mongolian prehistory, trying to understand when dairying first became important, and the population history over this time. So we've been doing a lot of ancient DNA work here. In collaboration with our partner Erne Mjagmar, we reconstructed over 200 ancient genomes and had genotype for the lactase persistence allele, and also looked for evidence of milk proteins within dental calculus to try to place this in time and space. One of our first findings was we were actually able to track when milk consumption first becomes important in Mongolia, and that begins about 5,000 years ago with the arrival of a group called the Afanaseva, which introduced dairy pastoralism to the region. And from there, it really spreads very widely and becomes the dominant subsistence strategy all the way to the present. We also were able to genotype individuals over 6,000 years of time, and we're able to show that the frequency of lactase persistence alleles is extremely low, and in fact, has really never risen above 6% during Mongolia's entire history. This is important because we can also show that over time, we get interaction with many different groups. And in fact, the European variant of the lactase persistence allele gets introduced many times into Mongolia by migrations of different herders, and it's never selected for. So this leads us with a kind of milk paradox. So Mongolia is, I think, a really interesting example of how you can have a strongly dairy-based society and not be reliant on lactase persistence, perhaps giving us some insights into how this may have worked during the early stages of dairy development. And so we've been trying to explore and try to understand if there might be other forms of adaptation at play here. And with this, we've been collaborating with Sonenkishin Solman, who's a nutrition scientist at the medical school at the National University of Mongolia. And we've been doing a lot of ethnographic work, partnering with herders across the country to try to understand, for example, the gut microbiome. So we have been collecting a lot of samples. So we've been doing lactase persistence genotyping, doing hydrogen breath tests for lactose digestion, and also collecting gut microbiome samples to try to understand if there's alternative forms of adaptation. And so we have partnered with more than 100 different households across Mongolia to investigate this problem, and are now in the process of analyzing some of our data. So this is very much like hot off the press, and we're still very new. But just to give you a sense of a quick peek at some of our gut microbiome findings. So one of the things that's really interesting, this is just a subset of the herders in our study. We see one thing that's really characteristic is we see a really high abundance of bifidobacterium. This is a bacteria in the gut that's known to be a milk-adapted bacteria. It's actually very high in infants, and typically is very common in young children and infants who are consuming milk, and then it decreases over time with adulthood. This is the typical range that you would see in adults worldwide. So typically less than 14% of the gut microbiome would belong to bifidobacterium, whereas we see a sizable proportion of the herders in our study have far more than this. For comparison, the typical average amount of bifidobacterium in the gut microbiome of infants is around 80%. So we see a kind of intermediate range here. And so we're wondering if some of these gut microbiome alterations might be the key to understanding how you can have a dairy-based diet in the absence of lactase persistence. And so one of the kind of questions we're trying to follow up on is, could there be multiple paths towards adaptation for lactose? Could one path that humans underwent be related to changing the human genome to adapt to producing lactase throughout the throughout adulthood, and perhaps a different adaptation is to adjust the gut microbiome to be more adapted to a high lactose load. And that may be actually also quite important and help explain this 4,000-year gap that we have between the origins of dairying and the earliest evidence of lactase production. And what's interesting is these two hypotheses, these two ideas, both are mimicking a similar feature. So either mimicking an infant-like state of lactase production or an infant-like state of the gut. In either way, there might be a kind of similar convergence here in terms of adapting to lactose. So with that, I will close because we'd really like to leave a lot of time for questions. So thank you so much for your attention. I just want to thank a lot of people who were involved in this study, and in particular, my two postdocs, Irina and Alex. So we'd be very happy to answer your questions now.
Great. Thank you very much, Christina. That was fantastic. Really exciting set of results. Thanks for sharing them. Yeah, we would love to see some hands raised. I hope that we have some questions out there. I understand that Bobby Schopler is here and has a question and is willing to ask. Thanks so much, Bobby. It's great to see you. Go ahead with your question.
Yeah, very interesting talk. Thanks for that. And I was just wondering, I've seen A2 milk in the grocery stores and have read some things that claim that A1 is a common cause of what we consider lactose intolerance, and that it's common in the new breeds like Holstein and Ayrshire, whereas A2 is more prevalent in goats and some of the old breeds like Jerseys and cattle that have been around for a much longer time, lower production, but also that A2 is most prevalent in human milk and goat milk and other types of milk. Have you seen anything about that or have any ideas about that?
So this is something we have not explored extensively. So I believe the A1 and the A2 variants, they're amino acid differences in casein. And it might have to do, I mean, some people do develop, some proteins can be quite immunogenic. And so sometimes people develop intolerances or to these different proteins. I know that also one thing that's important to know about human milk versus ruminant milk. So ruminants produce beta-lactoglobulin, which is a protein that has actually pseudogenized in humans. And so many people actually are develop an allergy to beta-lactoglobulin because we don't produce it as humans, and that can be a cause of intolerance to milk that's not related to lactose intolerance, but is a protein intolerance. And so I don't know if the A1 and A2 is similar in that sense, but one of the things that we are looking at now, which I do think is important, is how the milk is consumed. So especially very recently with the rise of kind of health consciousness around saturated fats, there's been a really large increase in consumption of fat depleted milks, so skim milks and low fat milks. But there's also a lot of evidence that suggests that consuming lactose in combination with high amounts of fat can reduce some of the effects of lactose. And partially what's going on there, at least what seems to be going on, is that high fat content dramatically slows down gut transit times. And this allows even though the amount of lactase production goes down over time and is very low in adults, you still produce a very small amount of it, even people who are lactase non-persistent. And so by really dramatically slowing down gut transit, that might allow enough time for the lactose load to decrease. So we're starting to really think hard about the role of fat and the different combinations of nutrients and how they work synergistically to aid digestion. Thank you for your question.
Okay, great. And I also want to make sure that we hear from Alex and Irina. Were either of you involved in any of the lactose work?
So yeah, I had the pleasure not just be involved, but also by the field trip involved me on a horseback riding adventure that I would like to share next time over beer. But no, we have been working with what was for me very fascinating is what Rutina was talking about. We in the Western society usually have a very particular way of consuming a lot of these like dairy products. We have usually milk, we put it in a coffee or in a cereal bowls in the morning, or yogurt maybe, and it's 3 different types of cheese. But if you go to Mongolia, dairy people have 15 different variety of consuming dairy products that you have never heard of until we actually went there and saw how they produce it. Like that they dry whey curds and then dry them in the sun and eat them as a rich sauce during the day as a snack. What we like, instead of having potato chips, they eat this whey curds and stuff like this. And I think also what we might need to understand a bit better is like Tina already put the fact, how much is the fat content might play a role. Also processing, what happens with their way of processing? Are they getting rid or changing the molecular structure of certain proteins by just like boiling the milk for a long time, or adding certain enzymes as natural occurring in soil cultures and things like this? So it's a very interesting. There's a lot of different aspects to look at. It's a very complex research field.
Yeah, thank you. And that relates to one of the questions in the chat from Sally Bornbush. Sally, are you there? And would you like to ask that? I know we have Elaine's hand up and some other questions, but just to kind of carry on with this.
Yeah, hi. I was really interested in whether you all were going to actually look at the microbes in the dairy products. There's some evidence in some other hosts that those microbes might actually prime the gut microbiome to better digest lactose. So I was just curious if you also have that, and whether you're looking at kind of the contributions of the milk or dairy microbiomes to the actual gut microbiomes.
And figure out how to raise my hand.
Yeah, that's a great question. Maybe I can start it and then have Alex follow up with some of the work he's also seen in the gut microbiome so far. So yes, so I just presented a narrow slice of it, but we wanted to have as comprehensive of a data set as we could and really view this from the point of view of ecology. So in addition to having the human gut microbiome data, we also have skin swabs as well, because when people are making dairy products all day, I mean, they're really, they also bathe in whey. So whey is used as a cleaning product for all their utensils, but also used as a bathing product. And so there's, and then the homes themselves are made out of felt and other porous materials like wood. And so this is something we were really interested in, and how microbes are cycling within this entire system. So yes, so for our study, so we have the human gut microbiome and skin swabs. We also have swabs of all the working surfaces and all of the tools that are used to produce the dairy products. We also have the dairy products themselves. So we have something like 36 liters of hundreds of different dairy products that we are slowly working on analyzing the microbial composition of. I will say it is challenging, because it turns out that when you have really high fat contents, it's very difficult to get a PCR to work. So if anyone has any suggestions there, we're finding a lot of our library constructions fail because the fat content is so high. So we're trying to optimize our methods there, but we are going after that. We have done some culturing work though, and have some genomes that we've sequenced with nanopore. And already we can see that it has some really interesting genomic features. So these microbes, which we call heirloom microbes, have not been studied. They are dramatically understudied, and they are different than the types of microbes that you find in industrial production. They're many of the same species, but they have different plasmids and different genomic capabilities, and we are extremely interested in this. We also did comprehensive livestock microbiome sampling. So we have we sampled saliva, fecal material, hair and skin, vaginal swabs, teat swabs from the livestock themselves. So we've really tried to get. We also have air quality, an air microbe filter sample. So we really tried to characterize this entire household as unit, and to see how microbes partition within it, how they cycle within it. And Alex has also been looking a little bit about some of the lactobacilli that we see in the gut microbiome. Do you want to say a couple words about that?
Yeah, so we see differences, but like at the moment we're still a bit on the early stage. We would like to improve the like everyone, we're mostly doing metagenomic analysis where you just shotgun sequence it and then try to assemble it, but that usually always has the issue that you can't really close the genomes. You don't know where the plasmids are, there if you have full plasmids and so on. So we're trying to, like Tina said, trying to get nanopore sequencing working, but that is at the moment challenging because a lot of the protocols are not optimized for this very complex media. And the culturing at the same time is also not trivial. So we're still like working on. But maybe another aspect I would like to raise, which I think for the part of evolutionary medicine might be something interesting, in from an ethnographic perspective, we were told from our the herders that we visited that these starting cultures are used every day there for doing the whole yogurt production and the cheese production, they are actually considered very valuable, as to extents that they're actually given as dowry to when you as a marry off into the next family, and as a woman leave the family, your family gives their local starter culture to kind of go to the next household when you find your own household, and passes it down, where then you again start your own culture, like your own like cheese production, yogurt production, by re-inoculating always the same starting culture. And especially what Tina said, we are sampling also the different households, the different utensils, and we want to also try to understand how stable is actually the starting cultures over time. Do we actually see the ongoing evolution, because this stuff reintroduced from the environments, going back from the animals? They're very clean people, but of course it's a different climate. They don't have hand sanitizers at every corner like I saw here in the US and in Germany, but everything is like much more in contact with nature. So it's first interesting to also just see how much diversity we observe and how much it evolves over shorter and longer periods of time.
And I think the other thing too, working with the herders on the issue of hygiene, which is something that's very important to them, because to maintain a dairy production, you have to have a lot of control over those microbes. And what's really interesting is they take a very different approach than is promoted, for example, through development agencies. So rather than trying to use sanitizers or chemicals or like bleaches or other things to drive down the microbial diversity to maintain hygiene, they take a completely different approach, and that is they encourage the dramatic growth of lactic acid bacteria. Lactic acid bacteria are great. They produce high amounts of acid, and acid is bacteriostatic or will kill most pathogens. So actually, by maintaining really high levels of lactic acid bacteria, they're able to maintain a really hygienic stock of dairy products. And so this is also something we're really interested in as a much more sustainable long-term way of maintaining high quality, healthful foods that don't require the inputs of fossil fuels and lots of chemicals that could be damaging to the environment. So working with microbes.
This is fascinating. Thank you so much for those perspectives. Next in line is Steve Stearns. He was having some trouble raising his hand, but he was the next one. Then we're going to hear from Elaine and then Lauren. Go ahead, Steve.
Yeah, okay, I've been muted. Great talk. I'm interested in the issue of why the ancestral condition was lactose intolerance, or to put it in another way, what are the costs of being lactose tolerant in a non-dairying society? So why did this happen? Our genomes have all the machinery they need to maintain the ability to digest milk as adults. And they have a rheostat that can set it to different levels, but it was turned off, and it appears to have been turned off consistently in ancestral populations. So what are the thoughts on why we were lactose intolerant in the first place?
I think for that, because a great question, we'd have to go deep back into mammalian evolution, because it's the ancestral mammalian trait, which is shared by all mammals. So it's believed to just be related to facilitating weaning and to force the offspring to stop consuming milk for the next generation. I think is really it's just an inherited trait from early in mammalian evolution.
Yeah, as an adaptationist, I don't accept the historical explanation. I want to know why physiologically it paid to turn it off.
It's a great question.
Maybe that's a great question, Steve. And another question I had related to those bifidobacteria. Is there a cost to having that high representation of that group of bacteria in the gut of these Mongolians? Why hasn't that been a general solution in other cases? Or I guess maybe it has, and we don't know about it. Is there a cost to that?
It's not known that there's a cost to it, but they are dairy specialists, so they increase in frequency when lactose is available, although they can metabolize other sugar substrates. And what we do see is that the bifidobacterial levels are higher in milk drinking societies in general. So populations that consume no milk will have some of the lowest levels, and populations that consume some milk will have higher amounts. You wanna?
Yeah, so and also one of the things is that we the samples we had, and Tina briefly showed in one of the plots, were all collected in the summer months, which is like usually from like June till end of June to mid of July. So you have usually a month, 6 weeks of real summer, or what we consider summer in the lower latitudes. And so we are now trying with collaborators here at Harvard to maybe have a complementary winter sampling, which is much more complicated because of the road conditions and traveling actually to remote places, but to have like comparative data to see how over the year it fluctuates, because at the moment we have this extreme peak, some of the individuals 50%, like almost looking like infants, and so we would like to see how much of a difference it does over the course of the year, if it's like cyclic adaptation, or if it's really always over the year so high.
Yeah, because milk consumption peaks in the summer, and then it drops in the winter, where meat consumption is higher. So there is a seasonal change that occurs.
Okay, thank you very much. And I'm sorry we lost Elaine. She had to run to another meeting. Lauren, would you like to ask your question?
Yes, please. So my question is there evidence of communities around the world consuming milk with other foods with different digestive enzymes and like good bacteria in order to compensate for the lack of lactase enzyme in their small intestine?
It's a good question. Not that I'm aware of. So most of the other populations that have been studied do have lactase persistence. Mongolia is really unusual in not having it. Can you think of any others? I can't think of any. I cannot think of any off the top of my head.
Okay, thank you.
Thanks for your question.
Yeah, thanks for the question. Does anyone have any questions about the first part of the talk? We're all focused on the lactase persistence and the gut microbiome. What about the oral microbiome? Any questions out there related to the first part?
I'll throw one out there just for fun. So there's the kind of classic look at dogs and amylase copies and how dogs have evolved to digest starch, but has anyone kind of paralleled the work you're doing, the other work you're doing in humans, with domestic canines versus other wild canines?
So do you mean like looking at the oral microbiome of canines?
Yeah, so oral microbiomes, gut microbiomes, and trying to see if, because there's also fossil records of ancient domestic dogs as well as wolves and other canids, and so looking at things like all of the interesting work that you guys are doing in early humans and ancient humans, but also extending that to canines.
I think I can answer that a little bit with, so we actually had a really funny project in our group a few years ago. Maxime Bory, one of our graduate students, worked on it. So one of the problems we have in the archaeological record is when we find preserved paleofeces, we can't always tell who made it. Because over time, it just becomes kind of smushed and deformed by the sediments, and so it can be hard to say. So we had this problem early on where we were trying to look at human paleofeces and look at microbial changes through time. And during one of our projects, we started to strongly suspect that we had dog poop mixed in with our human poop. So Maxime Bory ended up working on this problem and developing a computational tool, which he called CopperOID, which allows you to distinguish human poop from dog poop, because it turns out they look really similar archaeologically. And we found that actually one of our sites had a lot of dog poop mixed in, and then it made a lot of sense when we pulled it out of the study. So in that sense, we have looked at some ancient dog poop and some gut microbiome changes. What's really interesting there is that there are some parallel changes that we see in dogs that similar to what we see in humans. So what we see in humans is that with increasingly processed and industrialized diets, you get a simplification of the gut microbiome and loss of species in a shift. And we do see some evidence for that, or it does look like that similar process is happening in dogs. One of the problems that we have found, though, is that for the dog gut microbiome, so the only published data on dog gut microbiomes comes from like Purina studies. So they're eating like Purina chow, and there's very little work done on dogs that are fed like table scraps or dogs that are not consuming a highly processed refined diet. And that makes it a little bit difficult for us to understand kind of how it's changed. But it does seem that there's like a similar kind of trajectory that dogs undergo, similar to humans, with this kind of simplification of the gut microbiome with increasingly processed foods. There is also a canine oral microbiome study that was done a number of years ago now. Can you think of any more? I don't know. So in dogs, the amylase copy number expansion is in pancreatic amylase because they never develop salivary amylase, which in primates evolves due to actually a retroviral insertion into the pancreatic amylase gene. So it's a little bit different there.
Okay, thank you. We are almost at the top of the hour. I'm going to try to sneak in one more question. Cynthia, if you want to ask your question, please do. And Steve, if you're willing to type your point or your question into the chat, we'd love to hear it, but we're going to have to wrap it up after that. Go ahead, Cynthia.
Thank you. This is fascinating. It gives us a different perspective on both these topics. I was wondering if you had actually measured how much plain milk Mongolians eat. They have so many fermented foods or dairy products that, in fact, their exposure to the selective agent may have been very low.
So this is actually what we thought initially. So we have measured the lactose content of a lot of different dairy products because they extensively ferment their dairy. But we actually found that while the fermentation does reduce it somewhat, the lactose content is actually still pretty high in most dairy products. And in particular, we did a lot of work with people in Bulgan province, which is really like the horse milking region of the country. So in Mongolia, horse milk is used almost exclusively to produce alcohol because horse milk is low in fat and has proteins that are very hard to denature, and it has the highest sugar of all of the animal milks. In fact, it's about the same as humans. And so it actually makes a really nice alcohol if you ferment it. And but the lactose content, even in the alcohol, is quite high. So we measured this. It's a beverage called ayrag. And then even with the reductions that you see from fermentation in the summertime, because ayrag it spoils quite quickly. So people consume it during the season where it's available, they consume a lot of it. And its alcohol content can vary from 3% to 12%, depending on how it's made and the time of year, when exactly in the summer. But we were estimating that for some of the men in particular who consume large amounts of ayrag, so it's not uncommon for an adult man in Bulgan province to consume 250 grams of lactose per day during the height of the horse milking season through this fermented milk. So it's not a trivial amount. And most Mongolians begin the day by drinking milk. So with the first milking, having, they basically make a milk tea, which is essentially just tea brewed in milk. So really high levels of lactose consumption are consumed. And we have some numbers on that as well. We've done a whole nutritional survey, and the lactose intake is quite high.
Thank you. That's a surprise. Nothing
okay,
like
thank you. And yes, there's a comment in there. I'm glad to know that yak milk vodka exists. I have to say that caught in my ear as well.
Yeah, Shimin Art is quite good.
Irina, we didn't get to hear from you very much. I wonder if you want to try to take on Steve Stern's question. You probably can't see it from there. The question in the chat is, given your understanding of dental prehistory, do you have any insights into what modern dentists are doing wrong? You can also pass on it if you wish, but.
Yeah, I don't have any training in dentistry, so I'm not really set to say that. I would say clearing off plaque is really one of the best things you can do for dental health. So that is a pretty much the biggest thing that they do in industrial populations where we brush our teeth regularly. And
maybe you could talk about the toothbrushing effect that we do see though.
Yeah, so one of the biggest effects that we see when we compare ancient dental calculus to modern plaque samples taken from across the globe is that even though they do look really, really different, it seems to be just because of toothbrushing. So dental biofilms will grow up in a very specific way with certain taxa growing on the tooth surface and then certain taxa growing on top of those, and they form a really nicely structured biofilm. And when we brush our teeth, we disrupt the biofilm growth. And every time it's got to start from scratch, and it's got to start with those same taxa each time. So what we're comparing when we look at modern plaque and ancient plaque is the really early colonizing species to species that have grown through the entire biofilm stage. And so they seem to be more diverse, they seem to be more anaerobic, but really it's just because in our samples that we're collecting now, we don't give them time to get to that state.
That's interesting. Yeah, I just read an article in the New York Times about whether it's better to brush your teeth before breakfast or after. I'll let you guys do the research to find that article and weigh in on it. It wasn't very conclusive, by the way.